Genetically Modified Organisms Lab. Purpose and background. The purpose of this lab was to genetically modify a type of jellyfish bacteria, E. coli K12. This process was a genetic transformation. A genetic transformation is when genetic material carried by an individual cell is changed by new DNA being put into its genes. Things used were plasmids, arabinose operator, ampicillin resistance, and agarose. The agarose was made 3 days in advance before we did the lab because the agar needs to be dried out to take in the liquid transformation solution. Agarose contains ampicillin resistance and the arabinose operator. Agar gave hydrates to make the organism glow, and the GMO would grow without agar and luria broth, but not as much since they are food for the protein. Luria broth has minerals and proteins to make the GMO grow. The ampicillin resistance makes it harder to synthesize the cell wall in an E. coli. It is necessary, though, because it gives the resistance gene to the plasmid. Bacteria would die without it. But if they don't come into contact with it as soon as possible, they have the risk to die from it later. An arabinose operator was already in the green fluorescent protein gene, which is what made it glow. This is why we had luria broth ampicillin agarose versus luria broth ampicillin arabinose. It's the sugar that made it glow. Procedure. First, 2 microfuge tubes were labeled with plus P-glow and minus P-glow. Then, 250 microliters of calcium chloride were put into each solution. Calcium chloride is a positively charged divalent molecule that's attracted to the negatively charged DNA backbone. The calcium cation brings plasmid closer to the DNA backbone because of Coulombic attractions, and causes destabilization in the DNA molecule. Then, both of the tubes were placed in a styrofoam cup of ice water. After that, I took bacteria from the starter plate, which only had luria broth and agarose, by using a sterile inoculation loop. I used separate inoculation loops for each microfuge tube. I took bacteria from the plate to each microfuge tube and spun the loop to ensure the bacteria was in there. Then I moved the P-glow plasmid to only the plus P-glow tube because it was the only one that was supposed to result with genetically modified organisms. Then I placed both tubes back in the ice water cup for 10 minutes. Then I transferred both tubes to a 42 degrees Celsius water bath for a heat shock to happen. The heat shock will break bonds of bacteria, and when the tubes would be put back in the ice water cup, the bonds would be pulled back together. Then the 2 tubes were moved back to the ice water cup for another 2 minutes. I added 250 microliters of luria broth to both tubes of bacteria to give them nutrients. After incubating the tubes for 10 minutes, I put 100 microliters of plus P-glow in the luria broth ampicillin and luria broth ampicillin arabinose plates. 100 microliters of minus P-glow went into the plate with luria broth and ampicillin. After streaking each with different sterile inoculation loops, I placed them in a 37 degrees incubator and left them for 2 days before checking for the results. Results. The first plate contained luria broth and ampicillin resistance. It had no growth because ampicillin killed all the bacteria that could have potentially been there off. Also, there was no arabinose, which meant no glowing. It had minus P-glow and not plus P-glow. Plus P-glow would actually create genetically modified organisms, and minus would not. The second plate also contained luria broth and ampicillin resistance. The reason that this one actually had genetically modified organisms being grown in it is because it had plus P-glow instead of minus P-glow. There is no light because there is no arabinose, which is needed for the green fluorescent protein. The third plate had luria broth, ampicillin resistance, and arabinose. This plate actually had growth because it had plus P glow and ampicillin resistance. Because of that, genetically modified organisms grew, and because of the arabinose, they actually glowed.